Hi, Daniela. Can we start?
Okay.
Thank you. Hello, everyone. Good afternoon. My name is Arjun, and I'm going to host this session. And here today we have a speaker, Dr. Daniela Martinez. She's a evolutionary biologist and postdoctoral research fellow in the Department of Biochemistry and Molecular Biology at TGU. And she's expertise in the comparative genomics, genetics of populations, and ecology, phylogenetics, and bioinformatics. And currently, she's focusing on the molecular variant related to longevity and the cancer resistance across the species. And I hope this presentation will cover all the introduction section of the this useful tools like programming, like our R, analyze, and make a nicer thought from the different type of research data. So please welcome Dr. Daniela.
Hi, everyone. I'm gonna give a small introduction about what's R and how helpful it could be for us. So the talk will take about 30 minutes, and after, just feel free to ask me any questions. So in real life, we go to the field, we make observations, and we try to collect our data. So doesn't matter if it comes from the jungle, from cellular experiments in the lab, or even if you are working with databases already published. Doesn't matter the way or the source. My point is we are all gathering information. The question is, how do we organize, process, analyze, and graphically show to the world all the information that represents our work? In this opportunity, I'm going to talk about the different programming language, the advantage of R, how to install R and R Studio for beginners, a little bit about the language, the difference between the R Studio panels, and then I will go to an live session where I will show you the different ways to make plots, to import your data, and different types of data analysis. So what I want is to encourage you to jump into this super useful information tool, because there is always data that is waiting to be analyzed. So there exist different types of programming language that help us to process information. There are several ones. These are just some examples of it. So most of them will be different in how do they work. For example, Bash will be the top level language when interact with a terminal, and it's mandatory steps towards the data analysis if you are working with a cluster, for example, or with heavy data. While Python and R are one of the most common language used in data science. So my husband, for example, he's an astrophysicist and he's working mostly in Python, MATLAB, and Fortran, while I'm jumping between Bash, Python, and R. So what makes R so special? For a start, it's a free software environment used to do data analysis, statistical computing, and several graphics. So it can be used in several operating systems, such as Unix for the ones that use Linux, can be used in Windows and Macs, and is supported by the R Foundation for Statistical Computing. Also, R has more than 10,000 free packages, meaning that there is a lot of people around the globe that are writing codes and contributing to function, packages, and libraries to make our lives easier. So how to install R? It's pretty easy. You can go to the R platform and download the version that is good for you. And now I'm going to tell you how to install R Studio. But first, I'm going to tell you what's R Studio. R Studio is an integrated development environment for R. So in short, it's more a nice graphic environment than R, but it still depends from R. So it's easy to learn and work with, and it has more pretty visualizations. So today we will be working in R Studio. So before going to the Open Studio session, I'm gonna explain a bit about how the language works. You will see that it's very intuitive. So how to speak R? You can imagine that R is based in objects. Each object is like a drawer in which you keep information. So first, you need to name it. Like in this case, your object would be weight in grams. And this drawer will contain all the information that in this case will be the measurements. So if in R you do Control Enter, you can create your first object. And each time that you will enter weight in grams, you will have available this information. So there are common types of objects like vector, lists, metrics, arrays, and data frames. For example, a vector contains a sequence of data elements from the same basic type that could be logical,
Join the meeting.
double, character, complex, or can be a row. So for example, this vector length associated with our object weight in grams has 9 elements because it's the 9 numbers that we have gathering in the drawer that is named weight in grams, right? So if I want to know the class of elements, I could do class weight in grams. And in this case, it's going to be numeric because my values are numeric. But it could be also names or false true, for example. As the vector, you can make everything more complex and while adding different functions and arguments into your script. So RStudio has 4 basic panels. The consola, that is basically where you write the instructions that you want the program to execute. The source of the editor, that is where you're gonna write your code and everything that you wrote there could be saved as a script in a text file. And your code could be as simple or as complicated as you want. And you can open it and write it over anytime. The information area is where you can visualize your plots, the packages that you are using, the history, and other useful information. And the environment window, they can show you the different variables and functions that you are using in an open session. So don't be scared. I know that could be overwhelming this, but it's an amazing tool for analyze, organize, and visualize your research data. So now we're gonna stop to share this and we're gonna move to a live session. Just a second, I'm just sharing all the screen. Okay, everybody can see my screen? I assume
Yes.
that that's a yes? Okay, perfect. So normally the way that you work in R, you can work with different types of data. If you have data that is in Excel, for example, it's better to save it in CSV. For example, we're gonna work with this data that just have, like it's a basic data that is gathering from old and young mice that are divided in different categories. So if you see, this can be just a simple Excel file, but the difference is that this .csv divide each one of the variables and the data with commas. This is one basic type where you can just save your file. You can just go to Excel, save as, and you can put CSV because it's a common format when you're not going to have real characters that maybe you're going to mess up with your analysis. So to open an R session, you just need to have a text file that have the ending .r. So this is, for example, my script that I'm gonna present you today. So voila, you have already your R open. If you have R and RStudio already downloaded, they're just gonna open automatically. So The first thing that we need to do is set up our session and our working directory. So the session and working directory is where you have your data. So in this case, I have my files in the same folder than my script. So I could just choose source file location, and it's going to be here because all my data is in this same folder, right? Then after you set up your working directory, I'm going to teach you how to install some package. One of the package that are more useful and more downloaded from data science is ggplot. It's a super useful library that allows you to make like several types of plots. So for install packages, because you need to first download it, you can go to tools, install packages, and most of the package is going to be into the repository cron. So you can just put here ggplot2, and you can do install. I already have an install. So there is another complicated ways to install libraries, but this is the most easy way to do it. So after you download your package, you need to charge the library. So you put library and the name of your package. In any case, it's going to be ggplot. So you do Control Enter, and you can see in the console that it's going to chart the library ggplot, right? So I'm going to work here, but all the data that is going to be executed is going to be shown in the console. So if I want to know what the package actually does, I could do help package ggplot2, and here in help is going to show me like the documentation for all the arguments that are associated with this library. You can also Google how to work with ggplot, or who could work with other library in Google, and there is going to be a lot of tutorials. So ggplot has associated one types of data that are this one, that is already charged into the package, that the name is empty cars. This could be super useful for practice. So if you want to start with R and start to practice to how that works, you can just do empty cars, and it's going to give you all the information about different types of cars and different types of variables. So if I do head, the function head and empty cars are just going to be the top. I'm just going to see the top. And if I do plot empty cars, when I do plot empty cars, like the function plot, I'm going to see all the different combinations, right, of that my data has. Like this is just a basic way to see like how your data are distributed in the space. So we can see that maybe something is going on between the variable WT and MPG, right? Like we see like something is going on here. So the next thing is that we're gonna create a new object that is we're gonna name X, that is gonna contain the line, just the column WT, and we're gonna create another object that the name is gonna be Y, that is gonna contain just the column that corresponds to MPG. So if I do plot X and Y, okay, you need to first chart the objects. And if you do plot X and Y, you're gonna have your first plot, right? You already are plotting your data. If I want to do this more complex, I need to first introduce you to what the functions are. For example, the functions are the tools for complex operations that it perform a specific tasks. For example, a plot. Plot is a function, and there are mandatory functions associated that is going to be the variables X and Y, because the plot needs to have some data associated. So For each one of the functions, you can have also arguments that are the different options of parameters that are important for this operation. So if I put plot and I want to make them more pretty, I'm gonna add some arguments to these. And for example, I want them to, I want the dots to be bigger, or I want them to have a different shape. You see, these arguments indicates the shape of the dot. Or if I want to do 15, I'm gonna have different shapes. You see, sex is gonna be another super useful argument. And if I want to change the color, for example, I use the argument color and I could put green, it's going to change to green. So there are different arguments associated to your function that is going to make you make your plots and visualize your data in the way that you would like. So now I'm going to use the function ggplot. So this is with basic R, but if I use the same function with ggplot, that is actually the package that I'm using in this moment, I'm gonna see that I'm gonna plot which one? I'm gonna plot my data, that is 18 cars, and AES is a quoting function. So what I'm gonna compare? I'm gonna compare the WT, that is the x-axis, with the MPG, that is the y-axis. So I want to use the geopoint function, and I want them, the size of the dots, be equal to another variable that is associated to cosec. So this is another way to make different plots. So in this case, we are working with this type of data that is already inside of the package ggplot. But what if we need to work with our own data? Well, for import our own data first, we need to create an object. In this case, my object is going to call data. And my data is going to be in CSV. So I'm going to put the function read CSV, the name of my file. I want them to read the header, and each of the variables are going to be separated by commas. So if I charge my data, is going to be here already. Like it's going to be each one of the categories from all and junk mouse. If I use the function summary, I'm gonna have already some basic stats associated with my entire data. This is super useful because you're gonna have like the minimum and the maximum values, the mean or the median. That can be a basic thing if you want to see, like if you have outliers, for example. So if you want to check the distribution of your data, you can use your histogram. So I want to see what is the histogram between in my data old, just for the column old. So I do histogram and it's going to be there. So if you want to check how the functions works, you can do the symbol question and put the name of the function, and in help you're going to have here how this function works and all the arguments that are associated with them. Okay, so this is super useful functions that you normally use. So now we're gonna export our plot, and in this case I want to use the function bar plot. With the height I want to be associated with my data old, and the names to be associated with my data on each category. I want to also to have a title, and I want to name my axis, and I want them to be blue. So this is going to be used for the data all. So I could just do Control Enter in this case, and I'm going to have my first plot. If I do the same, but for the data junk, I'm going to have my second plot. And for example, if I want to make them in an array, because you want them to present that in a presentation or in a paper, you can use this function par, and I'm gonna tell them that I want them to be in one line, um, one line and two rows. Okay, so there are different functions super useful to work with. So here I'm gonna charge different libraries. So you combine libraries in order to work better and in order to fit your goal. So for example, these are libraries for color palettes. This is a library to make plots. And this is a super useful library to organize your data. So before, you see that my data was organized in this way. The categories, the values for old and the values for young, right? If I want to create a new object that is gonna be data GG, that contains information of data, but I'm gonna reorganize the data when I want the old and young be gathering in another variable that is gonna be named age, and all the variables and all the values that is gonna be in another variable that is gonna name number, I could just do head data G, and I'm gonna see how my data is gonna be different. You see? Now, this is a more easy way to work with your data. So now we can do exactly the same plot, but using ggplot, and you will see now you can have your different categories with different colors using like the age, old, and young. Okay, you can change the color palette, the labels. You can decide if you want to have a background or you don't want to have a background. So there are different functions that are super useful to work with. So these are, now I'm gonna give just quickly some examples of some comparative analysis that I used to make. So these are, for example, I'm charging a different type of data, okay, where I'm gonna have genes, I'm gonna have different control, different cell lines and my control line, and I'm gonna have data from gene expression or another measurements that you want. I'm gonna reorganize this matrix in something more easy to use when you are doing a script, that I'm gonna have just one variable that is gonna be cell lines. I'm just gonna have the gene expression is gonna be one object that is gonna contain all the values. So if I want to do my first statistical analysis that is gonna compare means, so I'm gonna create a new object that is gonna have the information from gene expression and lines that comes from my object data, and I want to do like the Wisconsin test, and I want to have an adjust method. So just gathering all just simple information and simple comments, I'm just gonna have my first data analysis from all my thousands of measurements that I made already. So if I want to save this, I could do write CSV, and I want them to save this object that contains all the results from my analysis, and I want them to the row names be true. So if I'm going here, data tree results, I'm gonna have my data already saved for the supplementary material, for example, for a paper. So this is super cool, and you can work with whatever you want. So now that we have all the statistics data, the question is, how do we plot it? How do we represent that in the space? So first, we can make a list that contains the different values that has p values adjust below 0.01. So we're gonna make a list about that we're gonna use later, that is gonna contain just this information, and then you can just plot your data, you see? So working in R is like make wishes. So I want my plot to be plot the x-axis. I want to be this variant. I want the y-axis to be this, and I want them to be filled through the cell lines. I want them to show the error bar. I want them to be a bar plot, but if I want a different plot, I could use just a different argument, and I can make them a violin plot. So just changing these arguments and plot them again. Well, I'm going to change my data, just a violin plot, when I could see like not just, I can see like actually how my data is distributed into the space. So as I told you, like this is like making wishes. So if I want to change the colors, for example, this is a super nice color palette that I really like. So I could go to Google and just put some of the palette that I want. And we have 6 variants. So we have the control line and 6 cell lines. So any one of you could tell me which one color palette you would like? Like anybody? I'm gonna put bigger if you want. Just can select one color palette. There are plenty of them here. So you can select one palette that has more than 6 colors, and you can just let me know. Someone wants to share with me the favorite color palette?
In the chat.
In the chat. I'm sorry.
Super Bloom.
But it shows Super Bloom.
Yeah.
Oh, I don't know. Super blue?
Yes, Super Bloom.
Where is super blue? Super bloom. Okay, super bloom. We're gonna put here a super bloom. Super bloom 1, right? Now we're gonna plot our data again. And they didn't charge this package, maybe because Super Bloom has just 5 colors. You need to choose one that has at least 6, because we have 6 types of data, or 6 variables. Another, can you just let me know? I cannot open the chat for some reason. Anyone? Another color palette?
How about Super Bloom 3?
Super Bloom 3. Okay, Super Bloom 3, perfect. So we're gonna change it to Super Bloom 3. If something happened here, I'm gonna receive an error, right? So I'm gonna have an error that is gonna be like insufficient values in the manual scale, because I put a palette that has less colors than the number of my variants. So I'm gonna use Super Bloom 3. And you see how pretty that looks like. So you can play with the colors. There are different packages and there are different websites that actually merge the different colors for you. Now that we have the plots, the thing is I'm gonna change it again to the bar plot. This doesn't have our statistical analysis. So we can actually, do you remember that before we make a list with the with the comparisons that actually has the p-adjust below 0.01? Now I'm telling them that I want them to plot the values that have the comparisons that have the p-value adjust below 0.01. So you see, doing these plots, you can actually made this happen. So this is going to be already a figure, a nice figure for your paper. You can do also differential gene expression analysis. There are several packages to do this. One of the packages that I use most is HR, but there is the dissect2. So there are several libraries to do this. So we're gonna have different samples, different treatments on time. We're gonna do the same. We're gonna group our factors. We can actually organize them, like we're gonna have 24,000 data, and there is in 12 rows. We can reorganize this data in a new object that is gonna name. We can clean the data. We can see the distribution of the data through an histogram. We can normalize all this data. We can plot like dispersion estimates. We can fit different models like the GLM to our dispersion data. And we can explore also differences between the libraries. For example, here we see that something is happening between this experiment and this one, right? You see that is separated into the space. So if I want to do like differential gene expression, we can make a contrast between C at week 1 and B week 2. So we can, what the good thing in R is when you create an object, we can use this object further in the analysis wherever you want. So we can, for example, have already our values of differential gene expression between this comparison of the data. We can summarize this data. So now we're gonna know that we're gonna have 300 genes downregulated and 400 genes upregulated. Simple plot to plot this information is this one. Use plot MD with the residuals of my analysis, and I want them to plot with the summary that I just made. So these can be also saved. So you're gonna have save these files, and you can have already these files here saved for your supplementary materials, for example, or to your wherever you want to put in the paper. You can also do pretty heat maps, gathering your information. And you can also make different comparisons across treatments. You can make like tests for normality to see if your data is parametric or not parametric. And then after that, you can choose the proper statistical test that you want to use. So there is another way that do a statistical analysis using the function from the same test. We can compare between just two groups of data. And you're gonna have here the plots. We can plot differences between categories. For example, here is really nice plot for difference if you have several categories. And then you can save these plots. You can do export, save image, and you can save this image in PNG. You can do vectorize if you want later to modify it. There is different format in which you can save your image. And finally, I'm an evolutionary biologist. So what I do best are trees. So these are different packages that I use to make trees. So this is data that comes from my PhD thesis, where I was working with longevity across vertebrates. So I was comparing mammals between sauros. Sauros is birds, crocodiles, snakes, lizards. So I was comparing mammals and sauros. And I was comparing like the body mass and the lifespan. So I'm gonna have data of body mass for each one of the species. And I was I have also the data from lifespan for each one of the species. So now here I plot my tree when I'm gonna have all the species, and the size of the dot is gonna be the size of the, I don't remember in this case what it is, but I think it's the longevity. So the size of the dot is gonna be related with the size of the value for each species. I could do it also for body mass, when I could plot I could make different plots. In this case, the thickness of the branch is going to be associated with the body mass of the species. For example, here, we're going to have the alligators, and here we're going to have the whales. We can do it in different ways, right? So the color of the branch is going to be associated with the value of the body mass. We can put it in a different direction. So this is a good thing on art, that you can just plot your things and organize them in different ways, and you can just put all the artists that is inside. And there are several like good examples to start in art to practice. I really like this one. That is the art graphic gallery that could help you to see which kind of plots that you want to make. For example, the scatter plot, and it's going to have different examples. And let's say that you like this one. You can go there and you can have different arguments and different ways to plot it. So it's a good way to practice with examples first in art, and then just pick up your own data. So this reference is really good if you are just a beginning in art. There is another shortcut because there is a huge amount of arguments, and it's really difficult to memorize. So if you haven't put your hands on art in a while, then you're gonna see, hey, how I do change the name of the axis, because like it's impossible to remember all the time all the arguments. So there is some sheets that are really good for remember all the functions or the principal functions and the principal arguments that you use to make plots. There is another group for the people that speak in Spanish, that the name is Latin R, where they make several courses and talks. And they are creating, like, I think, packages that are already made for people that doesn't speak English, that speak Spanish. And finally, this will be the end of the presentations. So thank you very much for this, and thank you very much to my laboratory people. So just feel free to ask me questions if you want.
Thank you so much, Daniela. This is a nice presentation. Session is open for question and answer. Please ask if you have any questions.
If you, I know that it's difficult to gather all this information right now, but if you want to start in R and you have that some errors that appear, you can just contact me and I will be happy to help you.
Hi, this is Yajing. That's great, and thank you for you organizing and presenting here, Daniela. Right, that's your name, and I'm very interested about the R analysis. I think this technique can combine so many softwares, preference, and together and to get a very beautiful, good data. And I might miss the first 10 minutes of your presentation. I guess the resources where we can get this software to use or make it more convenient for the new people in this field. I think that will be very helpful for us to start to use. I don't know if your lab have those of the links. We just click and then we can input our data and we can collect some analysis data or something.
Links? No, we don't have this in our lab. But if you have any questions about how to work with and you can start, I'm gonna post here the different links you can, for example, if you say like, I want to do this analysis, I want to be represented in this kind of plots, you can go to one of these examples and see how they distribute the information. And if you have any questions, you can just feel free to reach me, but no, we don't have, we don't have this kind of servers.
I see. I really want you can get this type of platform and to like us be very convenient to you, not always bothering you. That's
Yeah, but
it
it
will
would be a
be
very good idea. Yeah.
yeah, even we can pay a little, right? To not free to use everything. But it's if it's cheaper than other companies and we will pay a huge price to get out just one figure, I think that would be very worth in future. Thank you.
Thanks. How much does the subscription cost? No.
See, everyone's interesting about how to. So you will be have a huge market here.
I know, a new idea of business.
I like it. I like it. That's really very needed in every life, I think.
Okay, I think we don't have a question. No more. Okay, Danny, thank you so much. And thank you so much, Dr. Wang.
Thank you. Thank you both. Thank you for a very good organizing. Bye. Daniela, bye, Arjun.
Bye. Thank you very much.